Okay, so hi everybody. I'm honored to be here with you all today, and I'm Alexander Fisher. I'm going to talk to you about an ongoing modeling project of mine in Bayesian phylogenetics. So here's an outline of today's talk. First things first, I'll give some background about phylogenetics and talk about a motivating example via divergence time estimation. Then we'll unpack the big idea, the big model going on here, known as these local clock models. Third, I'll introduce my model and describe how it fits into this framework, the shrinkage local clock model. And this is where we'll spend most of our time talking today. And then I'll touch briefly on the inference machinery I use here, Hamiltonian Monte Carlo, followed by a brief proof of concept example to display how all these pieces sort of fit together. So first things first, here's a phylogeny, a small phylogeny. And a phylogeny is a tree that describes the evolutionary history of several taxa. So colored in blue at the bottom here, we have a few different tips and 3 tips. And so the number of tips in this tree is 3. And the way to think about each one of these nodes is that each one might represent a different species or a different biological specimen of some sort. It could be a viral isolate, or it could be a different type of mammal. And the green nodes, 4 and 5, are known as the latent or ancestral nodes. And typically we have information about the tips of the tree in the form of genetic sequence data. So you might think A, C's, T's, and G's, or perhaps codon data. And if that's the only kind of data that we have, in the absence of any other information, we can reconstruct a tree like this, where we have the relative age of each of these ancestral nodes. And so we might know that just based on that data alone, that node 5 here, the most recent, the common ancestor of all the tips, might be twice as old as node 4, the common ancestor to nodes 1 and 2. But if we want to place this tree on an absolute time scale, we need additional information. The additional information can come in the form of maybe a fossil record where there's some transitional form dated to be 5 million years old. And so now we know that node 5 should be 10 million years old if it's twice as old as node 4. Another way, we don't have to just use fossils, though. We could also imagine a situation where we have viral isolates for tips 1, 2, and 3, and they're all collected at different at different times. And if each viral isolate is sampled months apart or a few years apart due to the rapid mutation rate of some viruses, we might have a molecular clock whereby we can estimate time and determine the absolute time, place this tree on an absolute time scale. So this idea of placing the tree on an absolute time scale leads us to one of the questions that phylogenetics helps us answer, which is how long ago did some taxa diverge? And this is an important question because if we want to characterize the emergence of a virus, maybe it's spillover event from an animal reservoir, it's useful to know the time at which that took place. And it's also crucial to our understanding of how a virus flowed between different hosts. So in this example on the right, to draw your attention here first, this is an example from a paper by Borby et al. In 2014, where they studied the movement of the influenza A virus between hosts and placed them on this time-stamped phylogeny. So we have human influenza A, avian, equine, and each tree you see here is a different protein of the influenza A virus. So we have the hematogluten proteins, these HA proteins, as well as the Na surface glycoprotein and these other 2 phylogenies. And each one of these trees, just to build up some of the notation here, has anywhere between 92 and 355 tips. And each tip has a nucleotide sequence between 700 and 2,500 base pairs long. So within the phylogenetic framework, we collect all this genetic information into a matrix similar to this matrix of ACs, Ts, and Gs on the left here, where different base pair changes are highlighted in red. And so in general, we have an N, number of tip, by L, length of the sequence, matrix for nucleotide data. And the cool thing that Warby et al. Found was that the most recent common ancestor of different strains of influenza A seemed to cluster around a very small window of dates. So kind of this late 1800s, early 1900s, we see a lot of these most recent common ancestors. And one explanation of this is that there's a common progenitor to all of these different strains of influenza in each species. And one way to explain that common progenitor is that there was some global selective sweep that happened that sort of preserved the diversity that we see in each one of these different proteins. And this is in contrast to some of the previous theories that say there were a multitude of small selective sweeps across each protein individually. So this is just an example of some of the sort of cool, interesting questions that you can ask and learn about by estimating divergence times. So this is a small motivating example. And to unpack the model that they used to learn about divergence time estimation, or to learn about the divergence time of these various strains of influenza, they use this host-specific local clock model. But to unpack that, first I want to briefly tell you about the full-on relaxed local clock model. So this is a more general model. So to describe this model, consider this small phylogeny. And say we have our aligned molecular sequence data in some data matrix S, and we say that each site of that alignment evolves independently and identically according to some infinitesimal rate matrix Q. And this matrix Q governs the continuous time Markov process that leads to the rise of the data. This is sort of our data generative process. And what Q says is how an A becomes a T, a C, or a G, how a T becomes a C, a G, or an A, sort of how these transitions, transversions, substitutions all take place. And this is an important pillar of phylogenetic modeling because in contrast with distance-based methods where we might just compute pairwise distance between different taxa, by using this continuous time Markov chain model, we can account for things like back substitutions. It's just much more biologically tenable. It can account for insertions and deletions. And multiplied by this infinitesimal rate matrix is this RITI. And what this is a branch rate and a branch time. And I just wanted to point out that I've got this P is the finite time transition probability. And in the event that we're looking at nucleotide substitution, this would be a 4x4 matrix. But for codon substitution, it would be much larger. So we have, on each branch, we have this product of branch time and branch rate, and that product is the expected number of substitutions on a branch. So here, TI is actual units of time, like actual years, and branch rate is the number of substitutions per unit time. And so for that reason, it's often referred to as the molecular clock or the rate of evolution. So what I've just showed you is a tree that has a rate and time on every single branch of the tree. And the fact that there's a rate for every single branch gives this model the name the relaxed local clock model. But here's an idea, and that's the heritable clock idea. So some people call this just a regular local clock model. I think the term heritable clock is a little more apt here because the idea is, what if there's only a few locations on the tree where the branch rates actually change? And this is a form of model that Warby et al. Used. So the basic idea is that the rate itself is inherited along branches of the tree. The rate of evolution itself is inherited. And so we see that in this tree by seeing that branches with the same coloring, say the green branches here, all share the same substitution rate. And the authors of the previous example noted that the lax clock model led to model misspecification by falsely introducing rate heterogeneity on every branch of the tree. And they saw this with a number of simulated examples whereby the divergence time estimate was wildly off when the data was simulated under this heritable clock model. So the model they use prescribes each host, each influenza host, it's their own evolutionary clock rate, which kind of gets at this idea that maybe each host provides a different environment in which the pathogen can evolve, and the pathogen evolves at different rates within each host, at different speeds, so to say. And so what they do is they set out at the beginning to say each host has their own rate, and what do you do if you don't know precisely who should have their own rate and where the heritable clock should take place on the tree? Well, there are some models out there that do not presuppose the number of local clocks on the tree, such as the random local clock model of Drummond and Suchard in 2010. But they have a combinatorial approach to this estimating of local clocks that is a little bit slow on large problems, because they have to look at every possible combination of clocks on the tree. And if you think about it, each branch could either start a new local clock or not. So for a 10-tipped tree, there's going to be 18 branches in the tree. And in general, there will be 2 times the number of tips minus 2 branches. So since each branch can either start a new clock or not, that's going to be 2 to the 2n minus 2 possible clocks that you would want to try most, if not all, of those possibilities. So how do we overcome this this problem of learning about the number of clocks without doing it in a combinatorial way? Well, we use an autocorrelated rates model, which has the benefit of being continuous and not a discrete space we have to move over, which leads to a lot of natural ways to sort of move through the space efficiently. But autocorrelated rates models have some of their own problems that I'll touch on soon. First, just for the model itself, we say that each branch rate, Ri, is normally distributed about the log of its parent branch rate. That is to say that R2 is log normally distributed about R4, and with some variance sigma squared. Now, we reparameterize this model in terms of increments, saying that what we're really interested in is learning about the increment between branches, and from the stationarity of the normal distribution, we're going to get that these increments are normally distributed about 0 with some variance sigma squared. So we have these increments on the tree, and these increments are going to show us where a local clock is. Now, beforehand, we think to ourselves that there's small local clocks on the tree. So the natural thing to do in this Bayesian setting is to place a shrink prior on these increments. And so the question becomes, what shrinkage prior do we choose? And a first option you might think about is a normal distribution. Maybe most increments are centered on 0, but some move away. Another possibility is a Laplace distribution prior, where we have this exponential form for the increments, and this is sort of akin to the lasso in a frequentist regression problem. And the third option, which is the option that we choose, is a Bayesian bridge shrinkage prior. And I'll show you some pictures in a moment that I think illustrate the difference between this this prior and the other two better than I can explain with symbols. But to show you the symbol for a second as to why it's called the Bayesian bridge prior, is that it looks similar in form to this Laplace distribution, except that there's this exponent alpha. And this exponent alpha can be anything between 0 and 1. And 0 sort of corresponds to best subset selection, where we're just penalizing the existence of an increment that's non-zero. And 1 would bring us the Laplace distribution again. So we have this parameter that effectively bridges two different types of shrinkage priors that correspond to two different types of penalties in a regression setting. So onto the pictures of that sort of visualize the Bayesian bridge, so we can see just what's going on here. So on the left, I have this normal distribution in the green and yellow, and you see that it places not a lot of mass near, it doesn't place a lot of mass near 0. And in fact, it might be hard to tell an increment apart that's negative 0.5 or 0.1 from 0, or from 0.001. And you're going to see a lot of rate heterogeneity using this sort of prior. Taking a look at the Laplace distribution, we see that there is a lot more mass near 0. And we also have these exponential tails that take us down in mass towards 0 rather quickly as we deviate from 0. And this could lead to its own form of model misspecification, whereby now anytime we have a large increment between branches corresponding to a large change in molecular clock rate, we might be artificially shrinking that clock rate. So this is where the Bayesian bridge distribution comes in, and we have two very cool properties of the Bayesian bridge distribution. So the Bayesian bridge has these heavier than exponential tails, and this allows us to have rates that are really large if the data indicates. Secondly, the Bayesian bridge satisfies the oracle property, meaning that it will find signal in the data when there truly is signal, and that these increments won't be overshrunk. So this is a major boon over the other two priors, having this heavier than exponential tails, but it also leads us to some difficulties in learning about the rates. So we are taking this Bayesian approach to learning about all of these model parameters, and naturally we're going to use MCMC to Markov chain Monte Carlo to learn about them, but I'm jumping ahead of myself a little bit because I'll introduce the full posterior in a moment, but I want to touch on a couple subtleties of Bayesian bridge mixing. So it turns out that it would be nearly infeasible to set up this sort of prior and make inference under it, except for a mixing result, or a model parameterization scheme set up by DeVroy in 2009 that built off of some work from Mike West in the 80s and is well described in the Bayesian bridge paper by Poulsen et al. In 2014, where they say if we use this data augmentation strategy and think back to our rates themselves that are log normally distributed, the log of the rate is normally distributed, right? The rates are log normally distributed about their parent rate. Then the if we introduce this auxiliary local scale variable and follow through with our increment parameterization to have our increments be normally distributed about 0 with a local and global scale parameter, then when we marginalize over that local scale, over that nuisance parameter, we're going to recover our Bayesian bridge distribution that we desired. And this lends itself to a really nice Gibbs sampling of the global scale parameter, which would otherwise be a prohibitively slow task, preventing us from even using the Bayesian bridge shrinkage prior. But that's not the only problem we have, since we're this phylogenetic setting, because in phylogenetics, the likelihood sometimes has a heavy tail itself. And if we have a heavy-tailed likelihood and a heavy-tailed prior, we might be subject to an improper posterior, and we can't learn about anything. So using a regularization proposed by Aki and Mark in 2019, we place a regularization addendum to this Bayesian bridge shrinkage prior, whereby we essentially define a slab that we cut off the tails of the Bayesian bridge at, giving us a proper posterior. So to make an inference under this model, I'm going to set up the posterior that we want to learn about. And the posterior we want to learn about in the typical problem where this model would be useful would be the branch rates collected here as a vector R. We want to learn about the global scale. We want to integrate over this nuisance parameter, the local scale, and we probably want to learn about the phylogeny, the topology itself that connects all of the taxa under study. And we also want to learn about everything else being, the parameters that govern that continuous time Markov process, like the infinitesimal rate matrix entries. And we have some sequence data from which to learn all these things. So in traditional Bayesian formula fashion, we say that our posterior is proportional to the likelihood times the priors, and we're integrating out via MCMC this local scale parameter. And now I've just grayed out the bits to focus in on the HMC I'm going to talk about in a minute, which addresses how we specifically sample these rates. Because all the other quantities here, the tree and the model parameters of the substitution process are not the main focus, but are well discussed and well implemented in the B software package, where we also implement our MCMC method. So we're going to focus in on learning about these rates. And to do that, to make inference and do it in a scalable fashion, we use Hamiltonian Monte Carlo, but due to time constraints, I'm going to try to explain HMC just using this picture here. So, and I'm also happy to field more questions about this at the end of the talk. So what you're looking at is a typical 2D correlated parameter space, the typical set, which is the region of high posterior density that we want to explore after our Markov chain has converged to stationarity. And if we update one parameter at a time in this typical set, in this region of high posterior density, we're going to have a lot of proposals, right? If we just move in the X or Y plane, a lot of our proposals are going to be out of this region if we try to take large steps. So we're instead forced to make very small changes to move through and explore our posterior distribution. Now, HMC doesn't just move one parameter at a time, but moves all parameters at a time. And it does this by taking advantage of the geometry of this set using the gradient of the log posterior to propose large changes to all the parameters simultaneously and with a high acceptance rate. And so one point to focus in on in what I just said is that we're going to require the gradient of the log posterior to make all this happen. So a brief overview of how we go about getting that gradient, which if we did it naively and numerically, would make this inference machinery too slow to be usable, is we take a look at our gradient of the log likelihood and the gradient of the log prior. And it turns out that the gradient of the likelihood of the rates was just recently captured, just recently found to have a nice closed form expression that can be recursively calculated by G et al. In 2019. And all that we have to do is calculate a little Jacobian here to have the gradient of the log likelihood. And while calculation of this Jacobian is a little bit non-trivial, I won't bore you with the matrix algebra of that. And the gradient of the log prior, thanks to the nice normal prior on the increments given by de Vroy, we have an easy way to get at that too. A really nice simple closed form expression for the gradient of that normal distribution, right? So now for a quick proof of concept result as promised. So in this tree, looking at applying our shrinkage clock model to 42 different mammals And specifically, we're looking at a well-studied data set of the adaptive radiation of 21 rodents compared with 19 other placental mammals and 2 marsupial outgroups using the first 2 codon positions of 3 different nuclear genes. So what this leads to is 42-tip tree with a sequence alignment that's 2,422 base pairs long. And this is an interesting data set because originally published by Dowsery in 2003, they found that they were trying to see if the divergence time estimation of mammals of this mammal set was a product of strict molecular clock modeling. So they investigated to see if there was evidence for local clocks on this tree. And what they found was that their best supporting model under the maximum likelihood framework they used had 5 different molecular clocks. Subsequently, this data set's also been looked at by Drummond and Suchard in 2010, where they found between 6 and 12 local clocks. And reassuringly, we also find between 6 and 12 local clocks with a lot of the strong signals you see here perfectly replicating the signal they found in their paper. So this is a nice proof of concept that the model seems to be working, and that it may be useful to use these autocorrelated rates models and not be constrained to having small changes along the tree. And I'd also like to take a minute to say thank you to all of the awesome collaborators I've had the privilege to work with, who've put a lot of hard work into this paper and into this project. So Xiang, Aki, Philippe, and Mark. And I also want to thank you all for having me here today.